right, so welcome to Live Better Longer, Advances in Brain Health, a webinar brought to you today by the American Federation for Aging Research, Prevention, and the McKnight Brain Research Foundation. I'm Sarah Smith, the editor-in-chief of Prevention, a health and wellness magazine and website that's been around so long your grandparents might have gotten it. And really the greatest thing about my job is learning new things about health and new ways of thinking about health, which is why I love partnering with AFAR on our Live Better Longer series, where I get to chat with leading experts about their work and how it can affect all of our lives. And of course, these conversations are fascinating, but they are meant to be educational, and you should always talk to your doctor about things that might affect your personal health. And before we meet our panelists, I want to acknowledge the partners that made today possible. AFAR is a leader in supporting important research into health. Prevention, as I mentioned, has a long history of bringing awareness and thinking to a broad audience. And the McKnight Brain Research Foundation is a preeminent supporter of the incredible researchers who are investigating aging brains. And we're so fortunate for the work that AFAR and McKnight support. And I'm pleased to introduce two of those scientists to you today, both of whom have received McKnight's Innovator Award in Cognitive Aging and Memory Loss. Emilie Reas is an assistant professor of neuroscience at the University of California, San Diego. Hi, Emilie. Thanks for being here.
Hi, Sarah. Thank you for the invitation.
And Tara Tracy is an assistant professor at the Buck Institute for Research on Aging. Welcome to you too, Tara.
Thank you, Sarah. Honored to be here.
Thank you. Wonderful. All right. I'm so excited. We're taking the slides away so we can chat and talk about your work. So, I would love to start with an overview of the work that you do. So I'm hoping you can actually explain it maybe in two ways, or broad and then a little deeper. So first, how would you explain what you do to somebody that you met maybe at a high school reunion? So someone who's interested, but really isn't a scientist. Emilie, what would you say to them?
Yeah, so very generally, the aim of my research group is to understand the brain changes that occur both in normal aging and pathological aging. And we focus particularly.
I love Emilie, so I'm going to jump to you, Tara, and have you answer that question. And then when she comes back, she can finish.
Oh, there she is.
There she is.
Oh, did I drop out?
We lost you for a second, right after you said brain aging and pathological. If you could would you mind starting again?
Oh, sure. Yeah. So the aim of my lab is to understand the brain changes that occur in normal aging and pathological aging, with a focus particularly on Alzheimer's disease, which is the most common cause of dementia. And so to do this, we use a form of brain imaging called magnetic resonance imaging, or MRI, to identify basically markers of those brain changes. And a separate but complementary line of research in my lab is to understand the factors that contribute to those brain changes. So we focus both on non-modifiable factors, for example, older age, sex, genetics, but we also look at modifiable factors. These are things that you might be able to change in your lifestyle, your behaviors that could promote healthier brain aging. Things like health factors like hypertension controlling diabetes, or, for example, lifestyle behaviors like physical activity or adhering to a healthy diet. So the ultimate goal of our research is really to enable earlier detection of neurodegenerative disease and identify people who might be at greater risk for cognitive decline, and also to guide strategies to optimize healthy brain aging.
That's fascinating. And while I'm sure the person at your reunion who you'd be talking to would be like, so how am I not going to get Alzheimer's? I'm going to not ask that, and we're going to dive deeper in a minute. But Tara, can you answer that question too? The overview, what are you doing?
Great. Well, I'll start out similar to Emilie and say that my lab is very interested in in understanding more about what happens in the brain with aging and in disease when there's pathology. So we share that common interest. And so my lab is taking the approach of diving down into the cellular level to try to study the cells in the brain, in particular the neurons in the brain that are responsible for encoding memory. So my lab has a particular focus on trying to understand what are the bad things that are happening in neurons during aging and in disease that are causing memory loss. And so we dive even deeper into looking at the different proteins in neurons that are controlling the function of those neurons, and to see how they're dysregulated in disease. And so in the long term, we hope that this information will help us design therapeutics to try to ameliorate some of the memory loss that people experience with age.
Also fascinating. And again, well, I'm sure the next question would be like, wait, so I can take a pill. We will talk about that later too. I would also be curious if there's anything you would add to what you just explained to us, if the person you were talking to in an elevator or wherever is also a scientist. Is there, because I know we do have some on the call. Is there any little thing you want to add for those folks? Tara.
Oh, sure. I would add that I mentioned neurons, but really the thing that I'm most excited about that my lab is focused on is synapses. And the synapses are these tiny little structures on neurons that are responsible for neurons being able to communicate with each other in the brain. And this is really, I think, the center of where memories are formed. And these are the structures that are I think really affected in aging and disease, and then can drive this memory loss. So that's what I would add, and I can talk more about that later.
Super. I would like us to. Emilie, what about you? What else would you add?
Yeah, so just going into a little bit more detail about our methods, we actually use kind of a multimodal approach, integrating various forms of advanced MRI techniques. So we integrate conventional structural MRI with diffusion MRI, which allows us to measure brain microstructure, and also permeability MRI, actually a form called dynamic contrast enhanced MRI that allows us to measure blood-brain barrier permeability. And the blood-brain barrier is really the current focus of my lab right now. So for a little bit of background, we like to think of the brain as being protected from the rest of the body because it has this unique barrier around the blood vessels in the brain that protect the brain tissue from the periphery, and that protects it from things like toxins and other things. So we use this fancy form of MRI to actually measure the permeability or the leakage in the blood-brain barrier. And we're interested in that because vascular risk factors are known to contribute to Alzheimer's disease. And we think breakdown of the blood-brain barrier might be involved in that process. So we have a current study right now aimed at looking at basically how early in Alzheimer's disease the blood-brain barrier starts to become leaky, and also what factors contribute to its breakdown. And actually in my project supported by AFAR and the McKnight Brain Foundation, we're looking at the effects of peripheral inflammation on the blood-brain barrier and cognitive decline with aging. So that's a little more about the particular focus of our lab right now.
That's really interesting and helpful to hear. You mentioned, what factors contribute to decline, which I know is really of interest to so many people. I know that, of course, age-related memory loss is a scary idea for all of us. And, prevention and other media, what we are always doing is tell people, get tested early, be on top of this, don't be scared away from it. And early intervention is key. But sometimes is it opening a can of worms? Is that good advice? Do you think, in the broad sense of where we are with memory research, and how does that kind of fit into the work that you're doing, this sense of staying on top of it, knowing what's contributing or what you can be doing? Tara, would you start with that? That makes sense.
Sure. So what we know from the research in my lab and from many others is that often the cognitive decline is progressive, which is it happens over time and gets worse with age, right? So in a lot of the models that we use in the lab, so my lab uses mouse models of aging and disease. And so what we know from these models is that the synapses that I mentioned, synapse dysfunction tends to be one of the earliest things that happens in the progression. And so that's another reason that my lab is interested in studying synapses, because then, if we know what's causing problems at synapses that's leading to memory loss, then we can try to intervene early to try to repair synapse function and, have, better memory for longer. So that's the dream goal for us. So I do agree that if the earlier we can intervene in the process, the better. And, we think that figuring out those mechanisms is going to be really important first in terms of designing, interventions.
That makes sense. Emilie, how about you? How does your research fit into this, where we are with memory research?
Yeah, so I definitely echo Tara's point that, understanding the disease mechanisms are critical for early intervention, and we are almost there. I think, we are at really a turning point in research where, synaptic loss, like Tara is looking at, and vascular dysfunction, we're identifying that these pathways might be really great therapeutic targets. In terms of early screening, which is, I think, what you're curious about in terms of practicality, is, just like for every aspect of our health, I think it's very important to understand what one's baseline is when you're healthy early in life. Get tested early, yes, understand where you are cognitively so that you can monitor your change over time. So a problem with neuropsychological testing can be that if somebody gets tested, maybe when they're 80 years old, and they've never had testing before, they might perform very well on the test for their age compared to their peers, but they might be showing decline, relative to where they were 10 years ago. So it's really valuable to have, an individual baseline. All that said, we're at a very interesting kind of crossroads in research where we have really phenomenal biomarkers for early detection now. So some of our imaging biomarkers, and we have some really incredible blood-based biomarkers. You can just, hopefully soon in the clinic, you would be able to get a blood draw and be able to measure proteins that accumulate in the brain and cause Alzheimer's disease. So that raises kind of a conundrum. If we don't yet have effective therapies to treat neurodegenerative diseases yet, which we really don't, what is the value in getting screened either with cognitive tests or with these, accurate biomarkers, if there's not much we can do at this point to stop the disease? And I think there are still some advantages to getting screened and knowing that, for life planning purposes, or just reassurance maybe that you don't have the biomarker. But I think there needs to be a lot of education if we're going to start to incorporate, comprehensive screening, educating patients what actually is meant by these biomarkers. So
that's really helpful context from both of you. But the reality of where we are. And in fact, it's so interesting that you talk about, being at a crossroads and, in an exciting time. And I'm wondering if you could both help us understand, what do we already know? Where are we sort of right now when it comes to memory and memory loss, and what's happening in the brain? So that just to make sure everyone who's listening in sort of knows where we are as we then talk about what you're trying to solve and learn. Tara, do you have a thought on that?
What we know so far?
Yeah.
Well,
a little bit about how the brain works, and then so that then we can jump into, what you're learning.
Yeah. So maybe I'll get like a little bit more technical, but not, I'll try to explain it. So especially related to our research, we know that we know some of the things that are affected in the brain, right, in aging and disease. Some of the specific functions of the neurons that are affected. And my lab is specifically focused on a really important function, which is plasticity in the brain. And again, this is important because the plasticity in the brain is what's thought to encode the memories that we have. And this plasticity is happening at the synapses. And so in the formation of memory, you have a learning event, which promotes kind of a certain type of activity at the synapse. And then that activity causes the synapse to change and get stronger. And the stronger synapses is what we think is, causing the memory to be formed in the end. And so this process, this plasticity process, we know that it's impaired in aging and disease. What we don't know what really well, and what my lab is studying, is how is it impaired at the molecular level? Like, what are the proteins that regulate this plasticity mechanism at the synapse? How are they having problems to be able to do the plasticity? So yeah, I'll mention that.
That's great to learn to hear about plasticity. And Emilie, is there anything you would want to add to that? Or because I also want to ask you about inflammations more specifically. So
yeah. Well, I'm glad Tara took the cellular aspects there. She's the expert. So I'll just add a little bit more about generally what happens in Alzheimer's disease. So we know that there's an area of the brain called the hippocampus that is actually shows a lot of plasticity, like Tara was mentioning, and its main job is to basically encode the context of an experience. So say you meet somebody, you have a particular experience, the hippocampus kind of integrates all the sensory and contextual information from different areas of the brain and basically makes an integrated trace of that memory. And so by doing that, the hippocampus is involved in the initial encoding of the memory. And then over time, days, months, years, that memory becomes independent of the hippocampus, and the memory trace gets distributed more in the surrounding cortex, which is, the big bulk of the brain. And so what's interesting in the context of Alzheimer's disease is that the pathology and neurodegeneration in Alzheimer's disease first target the hippocampus and a region nearby called the entorhinal cortex. And so the hippocampus starts to shrink and it loses its ability to do that encoding process that I just described. So that is why one of the earliest symptoms of Alzheimer's disease is forgetting what you just did, because your hippocampus wasn't able to encode that memory. But that's also why somebody with Alzheimer's disease might be able to tell you with perfect clarity what they did 20 years ago. Because that memory is stored in a part of the brain that hasn't yet been affected by Alzheimer's disease. So that's kind of a more specific explanation of how memory is affected in Alzheimer's disease.
That's super fascinating, and also like I can almost have a visual of the memory moving out, and that makes so much sense when you explain it, because it seems so mysterious often to people, that my mother, my partner, whomever, thinks it's 25 years ago, but it makes sense biologically the way you're explaining it. Okay, so can you talk about inflammation though? You mentioned it, and I know that it's part of your study, and what do we know about what causes it, what's going on with it? But also I understand that it's sort of a difficult thing to study. And if you could talk.
Yeah, exactly. So just over the last, I would say, few years, inflammation has become a really hot topic in the world of just brain aging and Alzheimer's disease. We're now appreciating that a lot of the risk factors for dementia are related to inflammation. And what's interesting is that we see a lot of neuroinflammation in the brain of people with dementia. And the brain has these particular cells, they're called microglia. They're not neurons, they're a different type of cell. And they are responsible for responding to inflammation. And we believe now that in Alzheimer's disease, the level of activation can become aberrant. So they can become abnormally overactivated or underactivated. And one of the problems or the challenges to studying inflammation is that we don't really know the appropriate level of microglial activation in response to inflammation. We believe that it becomes disrupted at different points along the disease trajectory. So at a certain point, earlier or later, it might become overactive and then it becomes underactive. And so in terms of, a therapeutic target, that's really difficult because we can't just say, oh, give somebody an anti-inflammatory because some level of inflammation or microglial response to inflammation may be beneficial. So that's one challenge. Another challenge, which is actually the focus of my current project, is how inflammation in the body affects the brain. So there's actually this term that's been coined called inflammaging, which refers to the chronic inflammation that occurs with aging. And, that's thought to be responsible for a number of age-related diseases. And we're just now starting to think that inflammaging might contribute to the inflammation in the brain. And so kind of the hypothesis that our group is testing is that this peripheral inflammation in the rest of the body is affecting the brain via damage to the blood-brain barrier. So that's kind of a question we're trying to get at. How does all this inflammation in the rest of our body affect the brain?
That's fascinating. I can't wait to hear what you discover, because it sounds very, very like there's going to be some great progress there for all of our understanding. Tara, I want to know more about what KIBRA is, because I read a little bit about it when I was reading about your work, but I would love if you could explain to everyone what it is and why you're interested in this protein and its relationship to the brain.
Thank you. Yes. So we do have quite a number of projects right now working on this specific protein KIBRA. And there are a number of reasons for that. So I can first maybe just say a little bit about what it is and how it was first identified. KIBRA, the name KIBRA came from the fact that this is a protein that's expressed highly both in the kidney and in the brain. We don't know much about what KIBRA is doing in the kidney right now, but I've often been asked that. So hopefully in the future we can look at that. But we're really focused on what KIBRA is doing in the brain. And the reason for that is that it was first identified in a study on people where the gene that encodes KIBRA, there's a single a nucleotide or a part of that sequence in the KIBRA gene that's associated with memory performance in people. So if you have a better memory performance, it might have something to do with this one variant in the gene for KIBRA that you may have. And so that's how it first gained interest in the neuroscience community, that it's linked to memory. And then there's been studies since then showing in model systems, like mice, that KIBRA is required, the KIBRA protein is required for memory and plasticity in the brain, and that it's found at synapses. So it plays a role in the synapse in regulating this plasticity that I mentioned before, that's so critical for our memory formation. And this is specific in the hippocampus, as Emilie so nicely described. So this is plasticity in the hippocampus, where KIBRA is critical. And so what my work has done in the past, and also our current work in the lab, is to show that there's a KIBRA deficiency in the brain in Alzheimer's disease, and that this is associated with the memory loss in Alzheimer's disease. My lab now has some new work suggesting that it might be possible, if we can restore the KIBRA signaling of the synapse, that we can recover the plasticity of the synapse, even though you have the toxic proteins in the brain that are associated with Alzheimer's disease, the toxicity that we know is happening in the brain in these diseases. And so this is how we first gained interest in KIBRA, because we thought it could be its therapeutic target. But now the work that we have funded now from AFAR and the McKnight Foundation is looking more into how KIBRA modulates memory with aging. And there is also evidence from my lab and others that it could also play a role in cognitive aging and at the synapse. So we're going to be looking at that as well.
Interesting. It's fascinating how deep into a specific part of this you're going to teach us all about what's happening, in our brains. Emilie, could you share something, from your work that's an interesting finding, or where you are, the way, we just heard a little bit about Tara's, specific work?
Yeah, sure. I'd love to share just a little sneak peek for some of our blood-brain barrier findings. So this is still a very, this project is still in its early days. We just started this a couple of years ago, but we have some preliminary data from a small number of subjects that we actually presented at the Alzheimer's Association conference last year. And in this data set, we found that blood-brain barrier breakdown occurs in people with just mild memory loss in an area of the brain called the entorhinal cortex. And I very briefly mentioned this earlier, but it's right next to the hippocampus, and it's one of the earliest targets of Alzheimer's disease pathology. So that was really compelling that the blood-brain barrier starts to become leaky in one of the areas that we know also degenerates early in Alzheimer's disease. What's more is that when we looked at people who were still cognitively normal, so they didn't have any sort of memory loss yet, people who carried a gene called ApoE4, which is the strongest single genetic risk factor for late onset Alzheimer's disease. And I should note, this is not a deterministic gene. So if you have this gene, it doesn't mean you're going to get the disease. It just slightly increases your risk. So people who carry this gene also had increased blood-brain barrier breakdown in the white matter, which is, it's the structural connections in the brain that allow neurons to communicate with each other. So even without any overt cognitive decline yet, people who might be, in the early stages of Alzheimer's disease were showing blood-brain barrier damage. Then when we compared our measure of blood-brain barrier integrity to a measure of brain microstructure, which we can also get from MRI, we found that The greater the blood-brain barrier damage, there was greater microstructural damage as well. And this was also found in the white matter, or the connections in the brain. Interestingly, though, this association was only present for people who carried that ApoE4 gene. So this suggested to us that, blood-brain barrier breakdown occurs probably very early in the disease. And even before people start to show symptoms, it's associated with markers of neurodegeneration. So this kind of supported our hypothesis that blood-brain barrier dysfunction is possibly an early step in Alzheimer's disease pathogenesis, and possibly, we don't know if it's causative or not. We need a lot more research to look at that. So those preliminary findings are basically what prompted us to seek funding for these follow-up studies like that supported by AFAR and McKnight, to find out, what is causing this blood-brain barrier damage.
So can you tell me what you're hoping short and long term will come from? I mean, I have a sense of, where you're going with it. Is there an overview sort of thing, like in the short term, we hope to learn X and then that'll lead us to Y? I'd love to know that from both of you, for both of your work. But Emilie, if you could, continue with what you were saying with that context.
Yeah. So I think there are kind of two avenues that I'm hoping our research will contribute to. One is biomarker development. And by that, I mean developing measures that can help identify people who are at risk for either faster just cognitive decline with age or developing neurodegenerative disease. So right now, there are a lot of promising biomarkers out there, but, we're focusing on imaging biomarkers. And I strongly believe that the best detection approach for early diagnosis is an integrative approach. So I don't believe that, one of our MRI techniques is going to be the be all and end all for early diagnosis. But I do think that perhaps some of these measures that we're developing could be part of a comprehensive workup if a clinician thinks that somebody might be at risk and they want to further probe potential etiology of a disease. So I'm hoping, will contribute to biomarker development. At the same time, I'm hoping that some of our findings will contribute to an understanding of disease mechanisms and perhaps inform therapeutic development by identifying targets for therapies. So, for example, if we find that the blood-brain barrier really is a key and perhaps causal source of pathology in Alzheimer's disease, therapies that help repair the blood-brain barrier, could be a promising avenue. So that's hopefully where I see our research going.
Yeah, and that's such a great reminder too, that it's going to take work being done in a lot of labs and putting it together in interesting and innovative ways to find those therapeutics and solutions. Tara, what about you? Where do you see your work fitting in and where you want it to go?
Oh, sure. I think I should piggyback off of what Emilie said, which I thought was really interesting, her focus on biomarker, figuring out biomarkers. And I also wanted to mention, I don't think I've done this yet, is that we have also detected KIBRA actually in human cerebral spinal fluid, which is associated with cognitive decline in mild Alzheimer's disease cases. And so this is a kind of maybe short-term goal for us, is to really dive a little bit deeper into whether KIBRA could be a biomarker for synapse dysfunction associated with cognitive decline. And this could be, as Emilie said, an additional biomarker to help clinicians kind of figure out where people lie in terms of risk for decline or how far along they are in disease progression. And so this is actually something that my lab is also very interested in too. And maybe even Emily's lab and my lab could collaborate at some point for these things.
Tara, I was just thinking that. I'm going to email Tara after this.
I know. I mean, this is how science works. It's a great thing, to have these common interests. But in addition to the biomarker too, same as Emilie, we're all interested in therapeutics and how our research can be used to someday have a therapeutic. So my lab is inching forward on that in terms of the KIBRA project, in that we are designing a KIBRA-based peptide that we think in the future, in long term, could potentially be therapeutic and maybe make it into a human someday, which is very exciting. But right now we're doing all preclinical studies on mouse models. And the reason for this is to optimize the administration, see how well it works. We're hoping that this could be plausible, but we're still collecting data on that.
Interesting. You mentioned mouse models. Emilie, how does your lab work? What are you looking at and doing? It's not mice.
Yeah. So we work entirely in humans. We don't use animals or cells really. So we have a wonderful collaboration with the UC San Diego Alzheimer's Disease Research Center. And through that collaboration, we are able to enroll participants from their broader study. And these are typically participants from the community. Some of them have memory decline, some of them are cognitively healthy. And so they enroll in our studies, and then we conduct MRI scans. Through the collaboration with the ADRC, we also get a lot of very valuable other biomarkers. So for example, we might get measures of pathology from the blood or the cerebral spinal fluid. We get genetic information, and we also get results from cognitive testing. So we get a lot of really valuable data through this collaboration that we can compare against our MRI measures.
Interesting comparison. Tara, can you tell us a little bit more about the mice, like how you're learning from them, or?
Sure. So mice are so incredibly valuable for our research. We know a lot about their brain structure. It's similar to humans. They also have a hippocampus that has plasticity. That's and they also obviously express this KIBRA protein that we're interested in. And so my lab, we are highly skilled at working with mice and at looking at this plasticity mechanism in mice. And we also, the advantage of working with mice is that we can have models, like transgenic models. So these mice are genetically modified to try to mimic the human disease condition, because realistically, actually, the one reason mice are not good for studies of human is that mice themselves do not get Alzheimer's disease, or kind of these other dementia-related diseases that humans get. So we have to make transgenic ones that model the human condition better. And so we have these that we're using, and we can, we're testing these therapeutics on those models. And the other great thing about mice is that we can perform memory tests on them. Mice are actually have very good memory in general. So in our transgenic models that mimic kind of the conditions of Alzheimer's disease, for example, these mice have memory impairments. just as you would expect a human with Alzheimer's disease would have memory impairments. And so this is a valuable way that we can test whether a potential therapy can have a beneficial effect for memory in the brain.
Given what you've both explained about how you work and what you're discovering, this might not be a totally fair question, but I know that people always want to know, what about what can I apply to our lives? Are we at a point yet where you would be able to say that behavioral changes, or even just ways of thinking about our own brain from what you've learned in your work? Emilie, what do you think?
Yeah, I think we've come a long way over the last couple decades in really understanding what causes brain aging and what it actually is. So, I think for a while there was this misconception that, aging was almost like a disease where, you're destined to lose your memory and lose your functional ability. And we now understand that's not the case. So, if you look closely at a population, you see a wide range of trajectories of brain aging. Some people, live well into their 90s and still have, incredible memory. And so we're starting to really understand not only the factors that contribute to that variability, but also, some of the cellular mechanisms underlying those differences. And what's really important about that is that information is helping us to devise approaches to maintaining healthy brain and cognitive aging. And so, again, I don't want to be prescriptive here, but we do know that factors, such as diet, physical activity, adequate sleep, those are all really important. And some other things that people may not initially suspect, things like hearing loss is a risk factor. So making sure that you treat any hearing loss, social engagement, staying cognitively active, those are all things that we can do not only in older age but earlier in life to help optimize how our brain ages. And so I think we're like the research, not only from my lab, but, from others, is really sending a very positive and optimistic message that there's a lot we can do. And beyond just pharmaceutical interventions, I think there's a lot of very accessible ways that we can support our brain health.
That's great. Tara, is there anything you would add to that? Those great suggestions.
Yeah, that was great. And I think Emilie touched on this, but I would just echo that. I think it's good to keep in mind that there probably, at least I believe, there will not be one treatment that cures all memory loss, and whether it's just cognitive aging or Alzheimer's disease, or I don't think there's going to be one. I think it will depend on the person and their risk factors, and hopefully treatments will be designed together with these biomarkers we've been talking about, so that you can use the biomarkers to figure out what's the underlying problems, and then decide on whatever future treatments could be useful based on that. That's how I'm thinking about it looking forward, in terms of thinking about my own brain function even. Yeah, I would also echo, knowing if you have genetic risk factors that are associated with memory problems. But hopefully in the next decade, the whole field will be making much more progress on this. And hopefully, working towards different types of treatments, not just focusing on one.
Yeah, I think that's such a good point that you make, especially the more we learn about the complexities of just what you two are studying, much less what people in other labs are studying. And the more we learn about the brain, that of course, I mean, when you say it, it makes so much sense that of course there'll be more than one solution, the more we know. And I find that a very positive, hopeful thing to way to think about it, honestly, because it, like I said earlier, I mean, it can be so scary for people, but to know, oh, there's, a lot of ways in maybe. I'd love to know what excites you about brain research, even beyond your own work. What are you hearing and seeing and excited about? Emilie, what about you?
Yeah, so as I've already mentioned, I'm more on like the biomarker development side, but I think there are some really amazing cutting edge therapeutic approaches that are under development right now. A lot of these are still kind of in early stages. They're being tested in animals. I think there are some human clinical trials on the horizon, but just to kind of pique people's interests, there are some really amazing genetic and stem cell approaches. So for example, there are techniques that could theoretically allow you to go in and either, remove a gene that it causes a neurodegenerative disease, or introduce a gene that promotes, healthy brain integrity, healthy brain function, neuron growth, things like that. So those are some really cool genetic approaches that are under development. There's also some pretty remarkable stem cell research going on right now. So there's something called induced pluripotent stem cells, where you can actually take a cell from, you or I, and kind of reverse engineer it to become a stem cell. And then prompt it to develop into a neuron. So, theoretically, those approaches could be used to grow new neurons in neurodegenerative diseases like Alzheimer's disease or Parkinson's. And so we're still not there yet. I will echo, you can't go into your clinic and get any of these therapies. But these are almost, things of science fiction that I think are really close to becoming a reality right now. So I think we're at a really exciting time in neuroscience research.
That is exciting. Tara, what about you? What excites you?
So other than the fields that we're directly in, I started out my training actually in just studying neuron function in the brain, like just normal state, and also how neurons develop at early life. And so I still in my heart have really like that field. And I think I always think that the field of neurobiology in general, that we who study when things go wrong can really benefit from the field that's studying what happens under normal conditions, especially at the cellular level, because the more we understand about, how the brain happens in normal conditions, that I think that will really help, because that's also super important and not very well understood. I mean, we've got so many neurons in our brains and so many synapses, and it's just been, a very complicated organ to study. And so that the field of neuroscience in general, I get really excited about, and how that we can apply it to our research.
Yeah, that's so interesting. It's such a good reminder that just what's happening, to anybody is still kind of a mystery and will help us so much the more we know. Right. I love that. It's amazing, isn't it? What a mystery this central part of our lives remains. So I admire the work you're doing to help us chip away at all of that. We're getting close to our time. Before we totally end, I want to circle back to something someone asked. If we could expand a little bit more about knowing your personal baseline. Emilie, you mentioned that, and Tara, you also talked about that screening. Can you just mention or talk about how does someone do that? Is that something you can ask your doctor for? Like how do you do that?
Yeah, so if you're concerned about your memory, you can approach your doctor, and you would probably get a referral to perhaps a memory clinic or a neuropsychologist. And there are kind of standardized neuropsychological tests that they could administer. And these tests would assess various cognitive functions, such as your memory ability, your ability to do tasks that tap into, for example, executive function or attention. And using these tests, you would get, objective scores. And it would be recommended that, if you're concerned about your change over time, you come in at a regular basis and get maybe yearly or every 2-year examinations to see, the rate at which you're changing.
Okay, great. That's very helpful. Hopefully, I think, it sounds important to do, as you said before, you might think there might be a problem. So hopefully people will do that. Also pass that on to people that they know, who are, I know people in their 20s are like, whatever, I'm fine, but it's always good, right? To be aware of where you are. Yeah. Let's see, I would love to know actually if the work you do has changed your perception on aging. Just in your life, or, for people, Tara, has it affected your regular life, the work you do?
I think I would love to genotype myself for the Kibra variant. I haven't done it yet. But I am very curious now, so from my research, right? But I just think in general, like in my life and with the older people in my family and in my life, I'm just always so struck about the progressive process that happens, and how it starts out maybe not so bad, but then, slowly lifestyle is really affected by dementia and memory loss. And so, I actually, having that experience makes me want to try to understand it more in my lab, I guess.
Yeah.
I mean, the progressive nature of it, because a lot of the time when we design experiments, we tend to look at one time point, or I mean, that's how often in research design we're kind of limited, where we have to look at one time point. But trying to think about how in our studies we can look at multiple time points to try to look at the progression, and I'm hoping that kind of knowledge will help us understand the process more in aging in particular.
That's it. I hope you're able, to fulfill all those dreams of yours. Emilie, do you have an answer to that question? Because I also have another audience question to ask.
Actually, my answer was somewhat similar to Tara, so we can move on to an audience question if you'd like.
The question was about that, other researchers have observed that brain tissue and brain overall may age faster than other parts of the body. And is that something that you've observed? And do you have any thoughts about that? Is that related to inflammaging, or is that happening everywhere? What do you think, Emilie? Do you have an answer to that?
Yeah, that's a really good question. And I don't have a definite answer to that, but what we do know is that there is a lot of variability in both brain aging and systemic aging. And there have actually been a lot of efforts to develop, for example, brain age scores or biological aging scores. So you can submit a bunch of information about yourself, and then you'll get what your age is predicted to be. And there are, actual scores developed from MRI scans that are supposed to predict what your brain age is. And if you're higher or lower, then you're aging better or worse.
Are those real?
Sorry?
Are those real? Do they work?
I have confidence in the brain aging metrics to a certain degree. I think there's still a lot of room for improvement with some of our more advanced imaging techniques. So some of them now are still a little bit crude. I don't know as much about the general biological aging scores, but I don't actually know if we would say that the brain is more susceptible to aging. I think the brain, I think various tissues and organs are going to age at different rates depending on our exposures throughout life. So, if you're an athlete and you're really fit, you're going to have great cardiovascular health or whatever, versus somebody who's really sedentary. Different organs may age at different rates. So I don't think I have a very clear answer to that. It would be interesting though to kind of compare biological to brain aging and to really get at that question.
Yeah, and someone in the audience says, and in different cultures too. That would be super interesting. Lots of great ideas today for things that we want to know. We're almost at our time, so I want to ask, just one sort of last thoughtful question for you about, what do you think is an amazing or interesting thing about the brain? What makes you so interested in continuing to work on this area? I'd love to send us off with that. Tara, would you go first?
Oh, this is a surprisingly hard question because I get excited about everything.
That's why you do what you do so well.
Yeah, I mean, the most amazing thing about the brain is the computing power of it, and the complexity of it, both at the molecular level, at the cellular level, at the network level. Just every level of how the brain is constructed is absolutely amazing. And so, but obviously things start not working in aging and in age-related disease. So it's not, it's not completely, resilient with time. And so, it's a very challenging, the things that my lab and Emilie's lab are dealing with are very challenging things to figure out. But that I'm sure that's what drives both of us. Right?
Emilie, what about you?
Yeah, so I agree. This is a really hard question. But I would say my answer goes back to my original motivation for going into neuroscience, when I was an undergrad, which is just how remarkable it is that we have this relatively small blob of tissue, and it generates these just complex higher order functions like cognition, memory, emotional processing, consciousness, a self of identity, right? This is all housed in a pretty small blob of tissue. And paired with that, I have always found this to be kind of an almost amusing paradox, that our only tool to understand the brain is the brain itself.
Yeah.
And so, I mean, I always think of like an ant. Can an ant really understand its context in the broader world? Can the brain understand itself? And I don't, I like to think that we're going to be able to have that like meta understanding. But I don't know if we're ever really going to get there where we have a full understanding of the brain, because do we have the tools? I don't know. So,
I love that line of thinking. I think that's fascinating. And I think I appreciate you both putting into words, some of the things that make the brain so special and amazing and remarkable. And you both are amazing and remarkable in the work that you do and how you've explained it today. So I'm very grateful to you. And I know everyone on our call is, and I'm gonna, I think, have to wrap it up because we're just about at our time. So thank you both so much for all of this. Thank you to everyone who was with us today on the call and your great questions and thoughts. And We have another webinar on breakthroughs in brain aging coming up in June that I hope people will join us for. And of course, as always, you can support the wonderful work that these folks do and that, AFAR supports, at very simple at their site. So thanks everyone for being here. And thank you especially Emilie and Tara for your time and your insights. It's been really great. And I hope everyone has a great rest of your day.